There's all kinds of music, fast and loud, soft and slow.
Welcome to the rest of Chapter 8, in which we talk about gamete production, which is a vital part of sexual reproduction. Yeah, this chapter is all about the sexy process of meiosis. Of course, we're still dealing with single cells, so I wouldn't get your hopes up too much. In fact, no more music. So, meiosis is the process by which we create gametes. And gametes are the sperm and the egg. So gametes are sex cells. Meiosis is gamete production. So what two kinds of cells? I just said it. That's right. Sperm and egg. I'm glad you were listening just now. So what do gametes do? Well, if you haven't gotten this talk yet, allow me to help out. You see, when two people love each other very much, sometimes they decide to make a baby. Okay, never mind. Gametes are haploid. And as haploid cells, they only carry half the normal number of chromosomes that a normal cell does. This is vital, because gametes are destined to fuse together. So a sperm and an egg will meet, and the sperm will fertilize the egg, in which it fuses with the egg and donates its genetic material. And this is why gametes have half the number of chromosomes that a normal cell does. Because if it had the normal number of chromosomes, when a sperm would fertilize an egg and combine its DNA with it, you'd have double the amount of chromosomes. So, carrying half the genetic information allows a sperm to fertilize an egg and produce a functional organism. So, since we haven't used the term before, let's define diploid versus haploid. Which can also be described as in versus 2in, in which diploid is 2in and haploid is in. So, in a diploid organism, all of its chromosomes come in pairs. So, in humans, we have 46 chromosomes in a regular cell. So, those 46 chromosomes come as 23 pairs. And we number the chromosomes 1 through 22, and then the 23rd is labeled as the sex cells, which are XX or XY. Anyway, so because diploid chromosomes, a diploid organism, a diploid cell, has chromosomes that come in pairs, we call it 2in. In other words, there are 2 of in number of chromosomes. So our diploid number in a human cell is 46. 2in, 2 chromosomes for all 23. So 2 times 23 is 46. Making sense here. Haploid is in, meaning it does not have paired chromosomes. So a haploid cell is abbreviated as in. So diploid human cell has 46 chromosomes. How many chromosomes does a haploid human cell have? I'm listening. Yes, you over there. That's right. 23. Half the number of the diploid number of chromosomes. Now, it's very, very important that I use the terminology here, number of chromosomes. All right? We're not talking about the amount of DNA in the cell. Because all that DNA is organized into individual chromosomes. So diploid and haploid is the number of chromosomes. So here we go. Process of fertilization is when gametes combine, and so they combine their chromosomes. The sperm carrying 23 chromosomes and the egg carrying 23 chromosomes. And now we have 46 chromosomes, the diploid number. The single cell produced from a sperm fertilizing an egg, that single diploid cell, the first cell of a new organism, is called the zygote. So, the zygote is destined to undergo mitosis in order to produce a multicellular animal, if it will in fact become a multicellular animal. So, first cell undergoes mitosis to produce a multicellular animal. Let's mention some alternation of generations. Alternation of generations is the sort of state of cells as we go through a generation of organisms. So in animals like humans, it goes 2 in, 2 in. Meaning that you have an adult multicellular organism that has diploid cells, and then it produces haploid gametes, which then fertilize to create a diploid cell, which becomes a diploid multicellular organism. So this is seen easily enough in animals, and I just went through that. So the diploid multicellular animal undergoes meiosis, which produces gametes. And we have haploid gametes that then undergo fertilization that produces the diploid zygote, which then undergoes mitosis to produce the multicellular adult animal. Bam. So we're going to go through some words we're going to see a lot in meiosis. Homologous chromosomes is an important one. So the concept of homologous chromosomes is all about how an animal is diploid. So when we talk about this, we want to think about why the animal is diploid. It gets 23 chromosomes from the sperm and 23 chromosomes from the egg. I'm going to be using the human diploid number a lot. Not all organisms have 46 chromosomes, but let's just be anthropocentric here and deal with humans. So, 23 chromosomes from the sperm, 23 chromosomes from the egg. So, dad donates 23 chromosomes. Mom donates 23 chromosomes. Remember how I said the chromosomes are numbered 1 through 22 in the X or the Y? So, a homologous chromosome is essentially the pair of another chromosome. So a homologous pair is for, say, chromosome 1, the chromosome from dad is the homologous pair for the chromosome from mom. So, for instance, if you have one chromosome that carries the character eye color and, say, hair color, the chromosome from dad might code for blue eyes and black hair, and the chromosome from mom might code for green eyes and blonde hair. But because they both code for hair color and eye color, they are homologous. They code for the same characters. They're part of the same chromosome number. They're the pairs, the homologous pairs, the homologous chromosomes. Homologous chromosomes have sister chromatids that have the same length, centromere positions, and when you dye the chromosome, the same banding patterns. So, defining a character real quick, a character is a genetic category like eye color, height, hair color, so on. And a trait is the specific sort of the specific feature of that category. So if it's eye color, it's the actual color of the eyes. Blue, brown, green, so on. So the specific genes for that character. Blue eyes, green eyes, brown eyes, blonde hair, black hair, brown hair, red hair, so on. So traits are the actual gene for that, and characters are the category. Homologous chromosomes code for the same characters, even though they may have different specific genes for that character. Like dad having the gene for blue eyes, mom having the gene for green eyes. The character is eye color. The homologous pairs both code for eye color, even though they have different specific genes for that eye color. Bam. Easy. So, how does meiosis work? Well, meiosis is a two-step process, and it is incredibly creatively named. You see, the first step of meiosis is called, drumroll, Meiosis I. Now, what is the name of the second step of meiosis? I'm going to give you a minute because you may need to think this one over, mull it around in your mind. Okay, what's your guess? Correct! Meiosis II. My mnemonic for this is Meiosis I is first, and so it's 1. Whereas Meiosis II is second, and so it's 2. So you can take that one to the bank, helping you guys remember things. Aw, yeah. So here's a brief roadmap of the process of meiosis. But of course, we'll be going into far more detail, which is good because I know you're dying to know the details. So here we go. How does meiosis I work? It's kind of similar to mitosis, but a little bit broken. There's some significant differences that you must know, because there may or may not be a drawing question all about meiosis. So, since meiosis I is so creatively named with, 1, we're just going to continue that trend with prophase I, metaphase I, anaphase I, and telophase I. Amazing! All right, so you'll notice when we talked about mitosis, we had 5 phases of mitosis, whereas here we only have 4. And that's sort of to keep things simple. So in mitosis, we had prophase and prometaphase. In meiosis, we only have the prophase. So in prophase I, we pretty much have all the major landmarks of prophase and prometaphase of mitosis, with those significant differences I'm going to mention. So, if meiosis I is prophase, metaphase, anaphase, and telophase, with the 1 after all of them, who wants to take a wild crack at what meiosis II is? Well, I'm going to make you wait till we get there. So, hold on to that answer, and we'll confirm it when we get there. So, prophase I. Very important to discuss, because we're going to go into some big differences between mitosis and meiosis. So, just like in mitosis, our chromatin condenses into sister chromatids joined at the centromere. So we see chromosomes. But here in prophase I, the chromosomes arrange into homologous pairs. So the chromosomes coding for the same characters meet up with each other. They're like, hey, what's up? We have a lot in common. Why don't we hang out? And the other one's like, oh, that sounds awesome. Let's do that. And then they get together and synapsis occurs. Synapsis is, think about it as the zippering together of the homologous pairs. They join up into a strong, well, relatively strong connection with each other. So both chromosomes in the homologous pair join together and then zipper up. So that's synapsis. And then we call them a tetrad. So you may use the term homologous pairs, and you can use the term tetrad. I'll accept both, although tetrad is the technically correct answer. Ooh, technical correctness. The best kind of correctness. Anyway, we call it a tetrad. Well, tetra, 4. And a single chromosome is made up of how many chromatids? Sister chromatids. That's right, 2. And how many chromosomes are we dealing with? 2. So 2 chromosomes, each made of sister chromatids. Oh, 4 chromatids. Tetrad, 4. 4 chromatids in the homologous pairs. Now, a process called crossing over can occur during synapsis in prophase 1. We'll get into crossing over a little bit later, but basically in crossing over, one of the chromatids tangles over the other. So they come together and they're like, hooray, let's zipper together in synapsis. And it's like, whoa, oh no, and they tangled up. So a chromosome tangles with another. And we will talk a little bit later about what's so significant about crossing over. So, the nuclear envelope in prophase 1 then fragments. The mitotic spindle invades the fragmented nuclear envelope and attaches to the kinetochores of the tetrads. Now, it's important to remember that, so when you have your tetrads joined with the centromeres, I guess we'll just make my hand an unusually large centromere. When they're zippered together like this, kinetochores are available on this side here, and kinetochores are available on this side here, but the kinetochores where the chromosomes are synapsed together, not available. So the mitotic spindle attaches to the kinetochores on this side, and thus it's the homologous pair that's moved around by the mitotic spindle, and it pushes and pulls them around in the cell until we reach metaphase 1. So they're pushed and pulled towards the center, the metaphase plate. So here's a picture of prophase 1. This is some chromosomes that are synapsed, and this is crossing over occurring right here. So you can see they're tangled together. All right, metaphase 1. So you guessed it, metaphase 1, our homologous pairs are lined up at the metaphase plate. Homologous pairs are lined up. The homologous pairs are lined up at the metaphase plate. In mitosis, it was individual chromosomes that are lined up. Just all the 46 chromosomes individually lined up at the metaphase plate. In meiosis, it's the homologous pairs, the tetrads, that line up. So here we go. You can see our tetrads joined up, synapsed, and lined up on that metaphase plate. So it's worth noting that this example cell here has 1, 2, 3, 4 chromosomes. 4 chromosomes. So for this model cell in the picture, its diploid number is 4. That will come into play here in a moment. So now we hit up anaphase 1. Anaphase 1 is going to occur in a similar fashion to anaphase mitosis, except when we have this mitotic spindle shorten, pulling against these guys, we're not, I repeat, not going to break the centromere. The centromere is not cleaved during metaphase 1. So instead, we pull apart the homologous pairs. So the tetrad is split, and we have intact chromosomes moving towards the poles. So the homologous pairs, the tetrads, separate, and complete chromosomes move towards the poles. Sister chromatids joined at a centromere. So here it is. One chromosome moving up here, the other, and then down here, just like so. Minor difference, very, very important. And then finally we head into telophase 1 of meiosis. Telophase 1, we have the chromosomes at opposite poles, and cytokinesis occurs, producing two separate cells. And these are haploid cells. Remember when I just mentioned the diploid number of the cells in this example is 4? Well, take a look. This pole has 1, 2, and this pole has 1, 2. So we have 2 chromosomes in each cell. So those 2 chromosomes are the haploid number of chromosomes. So our 2 cells are haploid. The fragments of the nuclear envelope begin to appear, but we do not form an entirely intact nuclear envelope because we now go right into meiosis II. So here's our picture. Our chromatids are relaxing a little bit. If this were the end, they'd unravel into chromatin. So we've got a small amount of unraveling occurring. We've got nuclear envelope fragments reappearing. However, this is short-lived because we then go into meiosis II. So, review of meiosis I, the big landmarks. In prophase 1, synapsis occurs, and the homologous pairs are joined into tetrads that are zippered together. And then crossing over can occur here in prophase 1, where chromatids in a tetrad tangle together. In metaphase 1, homologous pairs, the tetrads, are on the metaphase plate. And in anaphase 1, it's those homologous pairs, those tetrads, that are separated into individual intact chromosomes. Then in telophase 1, we have 2 haploid daughter cells. All right. Mitosis versus meiosis I. Here's mitosis, and the big difference here you should look at is how these are not in homologous pairs. So when they line up individually along the metaphase plate, and so the mitotic spindle has access to both the kinetochores on either side of the centromere, so that in anaphase, when the mitotic spindle shortens, it cleaves the centromere and yanks these in two, so that they are individual chromatids. But here in meiosis, we have homologous pairs joining together. And then at metaphase 1, the homologous pairs, the tetrads, are lined up. And then anaphase 1, the tetrads are separated. And so where we started with 1, 2, 3, 4 chromosomes in the cells in meiosis 1 at its end, telophase 1, we have 1, 2 chromosomes in each. We're haploid. Bringing us to meiosis II. So, remember when I asked you what the phases of meiosis II were? Well, be prepared to have your minds blown. That's right. Meiosis II is prophase II, metaphase II, anaphase II, and telophase II. I bet you didn't see that coming. Telophase II is followed by another round of cytokinesis. So in meiosis II, we're now dealing with 2 cells at the same time. So let's go through meiosis II. And you are going to find this incredibly similar to mitosis. Because we no longer have homologous pairs, we are now dealing with individual chromosomes. So our centriole pairs duplicate. The mitotic spindle forms fragments of the nuclear envelope, because the nuclear envelope never fully reformed. Those little fragments that appear break down, and then the spindle invades that area and attaches to the kinetocores on either side of all the centromeres. And then we push and pull our chromosomes, our sister chromatids, towards the metaphase plate. That's like exactly the list of things that occurred in prophase and prometaphase of mitosis. So it's just here we're dealing with a haploid cell. So our spindle's forming, we have the fragments breaking down, we have 2 chromosomes here, and the spindle attaches to the kinetocores on either side of each centromere and pushes and pulls them towards the center, at which point we get to Metaphase II, in which we have the sister chromatids at the metaphase plate. Our intact chromosomes made of sister chromatids join at the center by a centromere. These are at the metaphase plate, just like in mitosis, except we're haploid. The microtubule is fully attached, and here we go. So 2 chromosomes here, 2 chromosomes here, and the next thing is anaphase 2, which happens exactly the same as it does in mitosis. In anaphase 2, we finally see the cleaving of the centromere. So here in anaphase 2 is where the centromeres split. See how I had homologous pairs separate in italics in anaphase 1, and here in big bold letters in anaphase 2 I have centromeres split. So making this kind of differentiation, being able to identify when the centromeres split versus when the tetrads separate, is might just gonna come up on the exam. Maybe there's a chance. So now our sister chromatids separate into individual chromatids, and they move to opposite poles. Now, it is worth mentioning that an individual chromatid here is still considered one chromosome for the purposes of diploid versus haploid. So, here in metaphase, this is one chromosome for the purposes of diploid versus haploid. And here in anaphase, this is one chromosome for the purposes of diploid versus haploid. So, the centromeres have cleaved in twain. They've split apart, and our individual chromatids are moving to poles, which leads us to telophase 2 of meiosis, in which the nuclei reform. So the nuclear envelope reforms around those chromatids. The chromatids are unraveling into chromatin, and we have 4, count them, 4 haploid daughter cells, and each daughter cell is genetically different from the parent cell. So, there we go. Bam. Okay, so let's mention crossing over before we wrap up this lecture. So, crossing over can occur during prophase 1, and it can occur during synapsis, in which point the chromatids tangle. Just like I said, one literally crosses over the other. Hence the name crossing over. A chromatid literally crosses over. Now, that site where the crossing over occurs, this little point right here, that's called the chiasma. So that is where the tangle occurred, the chiasma. So, metaphase 1 occurs as normal, even though we've got some tangling. And the big thing about what's amazing about crossing over occurs during anaphase 1. In anaphase 1, we pull on each homologous chromosome in that pair. And we pull on them in order to separate the pair. Well, unfortunately, those chromatids are tangled. And so you're getting a lot of force against them, pulling them. And think about if you have a tangle in some string and you yank really hard on either side. What's going to happen? That's right. That string is going to break. Well, the same thing happens with our chromatids. The chiasma is a site where the chromatids snap. And so everything past the chiasma, here, snaps loose from its normal chromatid. And now they're just hanging out. Well, once they're snapped loose, the homologous pair, the tetrad, separates. And as it separates, so here's my snapped loose part, and here's my original chromatid, and there we go. As it separates, we have this piece down here and this piece up here, and so I'm just going to stick to this chromosome. So here we go. This was from the homologous pair, and so this guy got his chromatid snapped off. This is the piece of the chromatid for the homologous pair that snapped off, and they're going to move apart. And what do you know? This snapped piece is now right here at the snap point on this one, and they're going to fuse together. So recombination occurs, and it occurs because DNA is attracted to DNA, and so the DNA on one chromatid is not, necessarily attracted to its own chromatid. It's just attracted to DNA. So when you have that broken piece of chromatid encountering the other sister chromatid that has space available, it just goes, hey, this looks as good as any spot, and joins up there. So doesn't care what piece it's originally from, and the broken bits automatically bind to the nearest DNA, which at this point just happens to be the homolog. And so the broken bits of those chromatids switch chromosomes. Whoop. And now we have our chromosomes even a little more genetically different. So here we go. Here's my site of crossing over. This is the chiasma. And we have labeled some genes on here. So these are some dominant genes, capital A, capital B, capital C. And this guy has some recessive genes, little a, little b, little c. So let's call like the C eye color, C for color. And big C there is brown eyes, and little c is blue eyes. And so we cross over here. And so this guy originally carried blue eyes, and this guy originally carried brown eyes. But once we get to anaphase 1 and we yank these guys apart and break the chromatids, they switch chromosomes. And now on this one, we have the dominant gene, the brown eyes gene. And on this one, we have the blue eyes gene. They've switched around. And so when we get to Meiosis II and we produce 4 cells, all 4 cells are genetically distinct from each other and the parent. So we have a massive amount of variation being produced by crossing over. So let's go ahead and stop here and pick up with why it's so important to have that massive amount of genetic variation. So see you in the next video.